The third or the fifth receptor, a nociceptor, right? This is the only class of receptor where the name doesn't immediately tell you the type of receptor it is. Nociceptors are receptors that sense pain. And so these receptors respond to excesses that might be received in the other receptors, right? Excess mechanical receptor, all right? Excess photoreceptor, right? Can promote a pain response. Again, the 5 classes, right? Mechano, thermo, photo, chemo, and nociceptor, right? They go as well with my mnemonic here, right? These receptors are also, most of them, susceptible to becoming adapted to the type of stimulus that they're receiving. Over time, if you continue to receive the same stimulus, this receptor will adapt to the point where it doesn't send that signal again. Right? So all of those receptors, with the exception of your pain receptors, experience adaptation. That's probably a pretty good thing that the nociceptors don't experience adaptation, because when you lie your pan on that hot stove, right, you don't want it to become adapted to it as you watch the smoke rise and your skin burns. Right? You want to be able to respond every time. Now, you may like the pain receptors and nociceptors to adapt if somebody's torturing you to try to find out where the last Oreo cookie went. But in most instances, the nociceptors are not susceptible to adaptation. Receptors can be simple. The simple types of receptors are involved primarily with tactile or mechanical stimuli, right? Pressure, stretching. Of these simple types of receptors, right, there are 2 main kinds. You either get encapsulated or unencapsulated. I'll look at a couple of examples of these. For example, an unencapsulated nerve ending, right? You can find these common in the epithelium, and they can sense touch. Right? They can sense pressure. Now, they can be a little bit more specialized. They can have sort of a disc shape that's associated with the end of this neuron here, called a Merkel's disc. But these guys are associated with mechanical impulses here, or root hair plexus. The nerve endings that surround this root hair here, are very simple, unencapsulated receptors. Right? Now think about this. These guys are very sensitive. Right? If an ant walked across the surface of your hand, you'd feel that, wouldn't you?
Mm-hmm.
Think of how sensitive you have to be in order to be able to sense something as small as an ant crawling across there. You could sense a flea jumping on you, couldn't you? I know that'd be true. I've been in houses where somebody didn't control the level of fleas that their animals have, and all of a sudden you're sitting there thinking, something's jumping all over me. And then you see all these little black dots over there. Yeah, it's a feeling. Right. Your root hair, right? When that mosquito buzzes by your skin looking for a nice place to land, you can feel it when it happens to hit one of those hairs. Right. You can have encapsulated simple mechanoreceptors as well, known as some of them known as Meissner's corpuscles. These guys are generally associated with the dermal papillae. Now remember, in lab, we're learning about the skin right now as part of what we're doing with muscles. Those dermal papillae, right, those hills and valleys, right, where the dermis and the epidermis meet. You can have encapsulated simple mechanoreceptors that are associated with that. Another form are Krauss's end bulbs, right? You find these guys oftentimes in the mucous membranes, like, you ever gone outside on a windy day and have that very, very small piece or particle of dirt hit your eye? Isn't that irritating? I mean, it is extremely irritating. And it's stimulating those Krauser end bulbs, right, that are located in the conjunctiva. So you find these guys in mucous membranes, the epineurum of nerve endings or nerve trunks. Some other forms of encapsulated simple receptors, right? The Pacinian corpuscles. Right? These guys are associated with receptors that you find in places like the interosseous membrane. Oh, interosseous membranes, where are those? Did we just do bones? Remember when we did bones, right? you have this connective tissue in between some of the bones, like the radius and the ulna here, or the tibia and the fibula. Right? This interosseous membrane in here has nerve endings associated with it. And those nerves in there, those Pacinian receptors, can sense stress. So when you jump down on something, right, you can sense that impact more, because these guys will sense that stress here in this interosseous membrane. Of course, you have a number of other receptors that play a role in that as well. Right? Other encapsulated nerve endings, Ruffini's corpuscles. Right? These guys are oftentimes associated with glabrous areas of the skin. Right? Those areas of the skin that have no hair associated with them. Right? And they can record pressure. They can record mechanical stress in those areas. Right? Moving to the next class of receptors, a little more complex than the very simple receptors, are your thermal receptors. Right? These guys exist as two groups of free nerve endings. Right? And they're there to sense a difference in temperature. Now, they don't work over the entire range. Right? You have two different types of them, warm and cold. Those warm receptors are responsive between 77 and 113 degrees. When you get above 113 degrees Fahrenheit, you now can no longer stimulate this warm receptor to be able to differentiate heat. Above 113, it feels just as hot as it does at 113. But what it does after that is it starts to stimulate pain receptors. Right? The same is true with your cold receptors, right? They differentiate at the lower levels between 50 and 68 degrees Fahrenheit, but when you start to get down below 50 degrees, right, you start to lose that ability to be able to differentiate the degrees of cold. And here too, again, you start to stimulate pain receptors. Now, both of these receptors, right, those cold and the warm receptors, they adapt extremely quickly. Right? So you can set a shower to be warm, but if you step right into there, it seems hot, doesn't it? But once you're in there a little while, it feels really good. Right? And so they adapt quite well. Right? Pain receptors, the receptors that don't adapt, right? Nociceptors. Now, these nociceptors, we don't have any of these nociceptors or pain receptors in most of the nervous tissue and in the brain. In fact, somebody can have brain surgery done on them, and they can lie there on the table, their brain totally exposed, and be totally conscious. You can talk to them while you do the brain surgery. And they've done that to try to identify what areas stimulate what types of responses too. Yeah, oh yeah, for sure. You can look. Yeah, that's wild, all right? You got your brain open and you're lying there and somebody's saying, oh, what do you think? I don't know. Oh yeah, that part of the brain stimulates that muscle. Right, these receptors, right, when they get stimulated, when tissues get damaged, right, when tissues get damaged, they burst open, right, they release their contents and they can promote a pretty violent response, but they increase the blood flow, right, and this damaged material for these tissues will cause pain in that area. And so that's one of the reasons why when you're experiencing pain, say sore muscles, because you just did more than you should have done last night when you were working out, right? Well, oftentimes we'll advise that you put some heat to that because it promotes blood circulation, which will help to wash away some of the damaged material. And now the pain can be reduced.
So why do they say ice it after injury?
That's to keep it from getting inflamed or swelling up. So you reduce the blood flow there if you don't want it to swell up. All right, I got a question for you. I want to know 3 large complexes that are associated with senses. Eyes, nose,
Brain?
ears, eyes, ears, nose. All right. All 3 of these large complex structures are associated with sensing our environment. All right, so let's start with it. Ears, nose, eyes, ears, nose. Right? Let's start with a chemical senses, which is associated with these. All right. Chemical senses, right? Chemical senses are associated with the sensation of gustation and olfaction. Your gustatory senses are your taste, right? What's olfaction? Smell. olfactory complex. So these these sensors that are associated with gustation and olfaction are chemical sensors, right? They respond to a molecule that's in solution. In order for them to respond, that molecule that will stimulate them has to be in solution. It can't be a free gaseous molecule, right? The reason you can taste food is because the molecules that are in that food have been dissolved and they're in solution. They may be dissolved in your saliva, right? When you smell something, right, you smell it only because it's been dissolved in your mucus. Right? So these chemical sensors only work when that molecule that's being detected has been in solution, right? Now, the senses of taste and smell, right, they often will respond to the same type of stimulus. Right. Let's take a look first at olfactory receptors, right? Here we have sort of an outline of an olfactory receptor. You breathe something in, it's in the air here, it is washing past the mucous membrane here that contains the nerve endings, right, that will pick up and respond to the molecule that's in the air here and cause a sensation in your brain. So this is picked up, moved through, right? Now, it goes through the cribriform plate when it gets to the brain. Do you remember where that is? You remember seeing this in the lab? That's why all these little holes are up here, right? Because those nerve endings, those axons have to move through there. Axons and dendrites have to move through there in order to be able to stimulate the cranial nerve. Okay? All right, so here we are, right? The molecule comes here, it gets sensed, and that stimulus is set up through the cribriform plate here, right, up to the olfactory bulb in the brain, right, which is responsible for being able to interpret this stimulus. Yeah.
So is that way if you're sick and you have excess mucus, you can't smell as well?
Yeah, because something is blocking these sensors here. Yeah, it gets built up. All right, so yeah, too much mucus is bad, right? You need some, but too much, right, reduces your ability to be able to smell. All right, so here, the sense of smell, right? In this sense of smell, right, the organ that's responsible for it is a yellow tinged patch of pseudostratified epithelium, right? It's in the upper part of the nasal cavity that covers the superior roof of this nasal concha. I got two questions for you here. Remember when we did the very first part of the lab, how did we identify pseudostratified epithelium? What did we look for? Yeah, those cilia, right? And look, there are cilia down here on these cells that line this. And why do you have cilia? To move stuff past, right? So you can smell better if you keep moving the molecules past, right? So that's by it's not just an odd coincidence that the cells that are associated with it have cilia associated with them so they can keep those molecules moving along past these nerve endings. Right. Also, the second question. This pseudostratified epithelium, right? It's on the superior roof of the nasal concha. Where is that? Yeah, it's got to be in there somewhere, right? Do you remember where the vomer was when we did that skull? On either side of the vulva are these small pieces of bone that stick out here called the concha, right? And on the superior side of this are the pseudostratified epithelium that's responsible for having the nerve endings for smell. Okay. All right. So here on the nasal concha, right? This contains the receptors, right? The neurons that are responsible for being able to take that message and send it up into the brain to tell you the type of molecule that you actually just breathed in, right? You have axons that are associated with the olfactory cells that extend through the cribriform plate. So we said that already, right? These axons axons. Let me see. Oh, not this one. Oh, wait a minute. That'll do it. All right. So they sense this and send that sensation up through the cribriform plate to the olfactory bulb. Right. In the olfactory bulb, it gets interpreted. What type of smell was that? Good, bad, right? Something that I should run from. Right. these neurons that are responsible for this, right, these neurons that are located right in this pseudostratified epithelium here are one of the few neurons that can be replaced. Right? These guys can die and be replaced. They have associated with them another cell called a basal cell. And if that neuron gets damaged or dies, right, that basal cell, and here he is sitting here, right next to the neuron, this basal cell will now be stimulated to differentiate from being a basal cell into a neuron, which is really cool, right? Because I know that all through your life in biology so far, everybody's always told you you don't get to generate any new neurons. Well, that's not true. Right? You generate new neurons in your nose if you need to. All right, now let's trace it again. Here, this molecule comes in through the nose, past the nasal concha, right, gets picked up by these neurons in here that are sitting in the nasal concha, the pseudostratified epithelium, and it gets passed up into the olfactory bulb so that you can sense it or interpret that sensation. When that gets picked up, right, there's what's sitting down here is a dendrite that's associated with this neuron. Now, let me ask you this. We talked about this a little bit. Neurons. Neurons have these structures associated with them, right? Axons and dendrites. So how many axons does any one neuron have? One. Only one axon. How many dendrites can a single neuron have? Lots. It could have one, but it could have lots. And so the dendrites are playing the role of taking this signal, right, of being stimulated and sending it into the neuron, right? So they are assessing. These are afferent dendrites. They are assessing the environment, sending the signal to the neuron here, which will send a signal out through a single axon, right, up to the olfactory bulb to be interpreted. Can receive multiple signals, but sends out only one. All right. So we said that in order to be able to smell, right, that molecule, that chemical has to be in solution in order to be able to stimulate this sensor. Right? And so that mucus is sitting there to help make sure it gets dissolved. The cilia are there in order to move it past more efficiently the neurons that are sitting in that nasal cavity. Now we know too that these types of sensors, right, that sense smell, right, and these are a chemical receptor, right? It's a chemical receptor because it has to be in solution. They are very quick to adapt, and every one of us who grew up in the Midwest know that's true. Your nasal or your sense of smell adapts really fast, doesn't it? Especially when they're fertilizing the fields. All right? After a while, you don't even notice it anymore. Right? And so these guys do adapt quite quickly. Right? But we have on the order, in humans, right, we can detect on the order of 10,000 different odors. Now, what I think is incredible is that we have on the order of 400 different genes that are associated with the sense of smell. These 400 genes are responsible for making 400 different proteins. The type of protein that gets made determines the type of smell that you can detect. Okay? And so each unique receptor, right, has a particular protein that's associated with it. So that receptor, because of that protein, can sense that smell. I think it's very cool. Okay? Now, when a particular molecule comes in, it'll oftentimes stimulate more than just one receptor. All right, so you have a mixture of different responses that are sent to the brain that it has to interpret here. We also know, too, that in the nose, right, we have both pain and temperature receptors in there, right? Because you can smell something to tell if it's warm, can't you? Yeah. This olfactory neural pathways, right, it goes through that cribriform plate. It makes it to the optic bulb here, or the olfactory bulb, right? That is associated with the first of 12 pair of cranial nerves. Right? So cranial nerve 1 is in the olfactory bulb, and is associated with the sense of smell. Right. When we get to our last section, you will learn all 12 pair of cranial nerves. But now you know the first one already. Right. All right. These sensory receptors for smell, right, they're extremely sensitive. Doesn't take much in order to be able to stimulate them. Right. When that signal reaches the olfactory bulb, right, that olfactory bulb interprets it and sends out a response. It can promote more than just one type of response. Right? And I think that you probably heard that your sense of smell can evoke memories way back in the past, can't it? So it can actually invoke memories. Right? And so there's a lot that's associated with this sense of smell, not just simply detecting that meat has gone bad. Right? In fact, you know too, certain odors, right, will cause a response of salivation. So there's a lot tied in to the interpretation of the sense that goes into the brain and the olfactory bulb sends back out. Now, we can, on occasion, lose our sense of smell. Right? It's called anosmia. Right? Oftentimes, it's the result of a head injury, or, as you outlined there, right, if you have a severe inflammation of the nasal cavity that impacts the ability of those neurons to detect and send those messages back in. Right? We know, too, that as you get older, your sense of smell seems to get lost or less sensitive. Right? And we suspect that that's likely because those basal cells, they're getting old too. And their ability to be able to replace a damaged old neuron is reduced as well. Right? And so it's like everything else when you get old, things just stop working. So my advice to you is don't grow old. Right? Now, I have a question for you too. We've all experienced in the last 2 years this very strange response to an infection. Right? What am I talking about? COVID. What have a lot of people argued that happens when they get infected by this virus?
You lose your sense of smell.
Yeah. And we thought that was a really strange response, that you lose your smell. Now, one thing we knew is the neurons that are responsible for smell, they don't have an ACE2 receptor on them. There is no reason for the virus to infect them. It doesn't have a receptacle there. There's no place for it to bind. And it doesn't. They don't get infected by the virus. So why do you lose your sense of smell? Well, those neurons need a lot of help. The support cells that surround these neurons do have those receptors. And when they get infected, and a big part of when it gets infected are the cells that are responsible for making those proteins, one of those 400 proteins that are responsible for smell. If they get infected and they die, and you lose the protein, well, the neuron can be there, but if the protein's not there, you can't smell. And so it was an interesting response that made us delve into further about how everything that this virus is capable of doing. And so that's how this virus is associated with this loss of smell. All right. Smell, taste, gustation. Right? We have over 10,000 taste buds, right, found on our tongue in the papillae. Right? Now, these papillae that we find on our tongue, there are three main types: fungiform, foliate, and valate. Right? And I'm not interested in you being able to distinguish one from the other. I want you to remember that these papillae are responsible for containing the cells that are able to sense taste. Right? We have a few of our taste buds that get scattered along the soft palate and the cheeks. Now, that seems strange, doesn't it? But you can put something right next to your cheek there and sense its taste a little bit, can't you? Yeah, that's strange. But some of them are associated with the cheeks and even the epiglottis. Right? And so, but most of them, right, are associated with the tongue here, along with the papillae. And oftentimes, right, they're not on the surface of the tongue, but they're within these little valleys in the tongue. They're sort of situated along here. The cells that are responsible for this, associated with papillae, are known as gustatory cells. They look like, and we'll look at this in the lab here in the last section, it's really easy to distinguish these guys. They look like little onions sitting among a bed of epithelial cells. They really are quite large compared to the cells that surround them here. Now, it's not just a single gustatory cell. Right? Each of these gustatory cells also have supporting cells associated with them. Oh, they have basal cells too. Why do you think they're there? What do you think they can do? Let me see, the basal cell associated with the neuron in smelling could replace that neuron, right?
Mm-hmm.
Well, they do the same thing for these gustatory cells. In fact, you lose gustatory cells at a very fast rate, and they get replaced. Now, I think it's interesting too. Look at what's extending on the end of these gustatory cells here, microvilli. We talked about microvilli a little bit, right? These gustatory cells have microvilli out here, and it's because a microvilli, it doesn't exist to move things past. A microvilli is there to increase surface area. And so if you can increase the surface area at the end of the cell here, you increase your ability to be able to sense the environment, right? That makes sense. All right, so these taste buds, right, made up of two primary types of cells, gustatory cells, which are actually the cells that are responsible for being able to sense that taste, and then the basal cells, which are effectively support cells. These basal cells will replace or differentiate into a gustatory cell when you lose that gustatory cell. Right. In fact, they'll do it about every 7 to 10 days. Right. A gustatory cell wears out rather fast. And so these basal cells are sitting there and they're differentiating on a fairly regular basis in order to make sure that you maintain those taste buds. Right. And again, recognize that these gustatory cells, the part that actually does the tasting is not here right at the surface of the sun, but it sits down in this valley, this papillae here. Right? And they're associated with these nerve endings here that send a signal back up this afferent dendrite in order to be able to be interpreted. We have three primary types of gustatory cells, and you don't need to remember all three types here. But one will release serotonin. This is a neurotransmitter. Right? Another releases ATP, and in this instance, ATP is also used as a neurotransmitter. Right? When we taste, right, we have 5 primary tastes that we can detect. Right? 5 sensations. And you probably are familiar with all of these, right? Sweet. Right? Things that have sugar in them. Alcohol tastes sweet. Right? Some of the amino acids will stimulate those nerve endings that sense sweetness. In fact, the lead salts you find in lead paint actually taste sweet. Now, you haven't experienced it much in your generation because in my generation, we were committed to getting all the lead paint out of homes because children kept flaking it off and eating it. And now you understand why they would do that. It tastes sweet. But now these children would have high lead levels, right? And so we've spent a lot of time trying to make sure all that lead paint was removed. If you move into a very old house, right, or you're in an old house where the paint hasn't been removed or treated, be aware, right? It could contain lead paint. Very popular form. Lead gives you a nice intense color. As a paint for color, it was really good, right? But it's toxic. Right? No, it's sour. Right? These sensors that's stimulated to give you the sour sensation, they are responsive to levels of hydrogen ions. Right? So something that's acidic will taste sour, right? Because it's a function of the hydrogen ion concentration. So acids will stimulate this. Right? A lemon. Right? My granddaughter is the only person I know who actually prefers to eat lemons. I don't get it. Right? Salt. Right? Metal ions will stimulate this sensor. Right? The sodium and sodium chloride, that's a metal. Right? It dissociates completely when you dissolve this. Right? And that metal ion here will stimulate this, and you get to sense that salt taste. Something that's will stimulate the bitter sensors are alkaloids. Caffeine is an alkaloid. Now, most of you might say, well, I've been to Starbucks 100,000 times, and everything I've gotten tastes extremely sweet, even though they say the caffeine is way up in there. Well, caffeine by itself is extremely bitter. I purified some and tasted it once, and believe me, it is extremely bitter. Right? But when you mix it with a whole bunch of sugar, you just don't sense it. That this last one, the umami, right? Amino acids like glutamate aspartate will stimulate this sensor. that's what gives a nice, good steak its nice, great taste. And we've figured out how to artificially produce this now, so you can actually just spread a steak over your salad if you want. Right? Just like the sensors that are used for smelling, in order for the sensors that sense taste to be active, right? That chemical has to be dissolved. It has to be in solution in order to be able to taste it. Now, one thing we recognize about taste too is that 80% of what you sense as taste is actually the result of smell. Now, you know that when you have a cold, things just don't taste good much anymore, right? And so smell plays a huge role in your taste. Right? These receptors too adapt extremely fast. And so if you want to know how something truly tastes, right, when you first pick it up in your spoon or in your glass, pay attention for the first 5 seconds, because after that, your senses have started to adapt already. So the true taste is the taste you get in the first 5 seconds. Right? After about 1 to 5 minutes, completely adapted. You can't really tell the difference anymore. Right? There are disorders that are associated with taste, but they're not very common. Right? People who receive radiation treatment right around the head of the throat area, they will oftentimes lose their sense of taste or have it depressed a little bit. We've recognized that if you provide zinc supplements to these patients, right, it helps to reduce the impact of the radiation treatment with respect to its impact on taste. Now, folks have said, right, that they also lose their sense of taste from a COVID infection too, right? But it's probably mostly the result of the loss of sense of smell. Right. Now, why? Why is it tough to lose your sense of taste? Right? Well, there are 3 nerves that are responsible for sending that impulse to be able to sense taste. Right? Facial, basopharyngeal, and vagus nerves. Nerves 7, 9, and 10. 3 more of the 12 pair of cranial nerves that we're going to learn. Right? So it's unlikely you're going to totally lose your sense of taste, because you have to impact all 3 of these nerves. All right. Again, adapts rather quickly. All right. Here, the different types of taste, right, they have different thresholds for being able to depolarize that sensor or that nerve, right? Bitterness, those bitterness sensors are the most sensitive of them. Right? It takes the least amount of stimulus to be able to depolarize and send a signal from those nerve endings that sense bitterness. Right? Salty and sweet, it takes the most. So these guys need more in the solution there in order to be able to fully stimulate them. Right? That takes us to the point of being able to talk about hearing. So, because I want you to hear the rest of what I want to tell you, I'm going to let you go. And we will do hearing and sight the next time we get together. All right.